Dear students, it's lecture about connective tissue. In human body present more types of connective tissues which developed from mesenchyme. Mesenchyme is connective tissue of embryo. In human body present more subtypes of connective tissues which represent special functions and special compositions. There are blood connective tissue propria, which include fibrous connective tissue and specialized connective tissue, cartilage, and bone. On its slide, we can see classification of connective tissue scheme. Here are four types of connective tissue which represent special characteristics and special functions. About characteristics of connective tissue. In connective tissue present more part of extracellular substance and appositive of epithelium when not present extracellular substance. Here in its type of tissue present general part of extracellular substance and cells. In general, extracellular substance include two main parts, fibers and amorphous ground substance. Its composition present in all types of connective tissues except blood. In blood not present fibers and amorphous ground substance not present too, but in blood present another extracellular substance which has name plasma of blood. On its lecture, I will represent to you composition of extracellular substance more because all types of connective tissues except blood include extracellular substance. About fibers. Fibers to divide on three main types, collagen fibers, elastic fibers, and reticular fibers. Collagen fibers include special molecules of collagen fibers, and very important, collagen fibers form very strong bundles which has size by diameter 0.5 to 15 micrometer. Reticular fibers are similar to collagen fibers, but include some specific composition of molecules, highly glycosylated and form delicate silver staining networks. And it's very important characteristic of reticular fibers, which use in investigation of composition of reticular tissue. Reticular fibers include in reticular tissue, which stay more in organs of hematopoiesis. It's red bone marrow, spleen, lymph nodes, and next type of fibers is elastic fibers. Elastic fibers consist of specific amorphous protein, elastin, and protein microfibrils. Include in specific ground substance of elastic fibers. Diameter of elastic fiber near from 0.1 to 10 micrometer. And elastic fibers maybe form three subtypes. More young composition, it's specific elastic fibers, oxytalan elastic fibers, alanin elastic fibers, and methyl elastic fibers include near 90% more ground substance and 10% fibers. On its picture, you can see different specific staining of collagen fibers and elastic fibers. First part of its picture shows staining after hematoxylin and eosin. In its case, collagen fibers takes more eosin elements and provide a red or dark pink color. Elastic fibers stay more black color after specific staining or dark brown color after specific staining of orthine. It's very good scheme present also in your textbook, which represent composition of collagen fiber. And you can see collagen fiber includes some elements and form step by step. The first element on its picture number 1, it's a molecules of collagen which form tropocollagen, and tropocollagen together form parallel layers, and in result visible special striation of collagen fiber. Next, tropocollagen collect and form fibril. Fibril number 2 and then number 3, it's a more strong composition because include more parallel form collagen fibers. Next step formation, it's a bundle of collagen fibers. Bundle of collagen fibers include more sub-collagen fibers which surround of elements ground substances and in result form very strong structure. Specific characteristic of collagen fibers is strong, not elastic, and staining by eosin and form acidophilic color in hematoxylin and eosin. It's dark pink color or red color. On its slide present photo from histological specimen after staining of orthine, and orthine represent elastic fibers in slide, and elastic fibers after staining of orthine stay dark red, brown color, and form specific network, but you see it's a very thin fibers, appositive of collagen fibers, and not strong, more flexible. The next part of extracellular substance is amorph ground substance. Ground substance consists of mostly glycoconjugates, which form two main classes, proteoglycans and glycoproteins. And composition and amount of glycoproteins, proteoglycans represent specific characteristic of ground substance. The next part of connective tissue propria is cells. Connective tissue cells subdivide on two main groups, fixed cells and wandering cells. Fixed cells, it's cells which form of connective tissue and stay, not wandering, not migrate, stay more in one place, maybe form vacuums and provide formation of connective tissue elements, extracellular substance. Fixed cells include main cells of connective tissue, fibroblasts, and all groups of fibroblasts. It's different to start from mesenchyme cell in embryonic period, and next generations, next differentiation lines, it's progenitor cells of fixed cells, fixed fibroblast, is prefibroblast, fibroblast, and final stage fibrocytes. Fibroblast predominant cells in connective tissue propria, and itself provide and form components of extracellular matrix. I repeat, which is this? It's fibers and ground substance. Next type of fixed cells is reticular cells. Reticular cells produce reticular fibers and present in reticular tissue. Reticular tissue includes in red bone marrow, spleen, lymph nodes. On its slide, present scheme of fibroblast. Its large cell, central position of nucleus. Nucleus is euchromatic, very active, light staining nucleus. Next, inside in cytoplasm present more organelles of synthesis, rough endoplasmic reticulum, Golgi apparate, mitochondria, which include in process active energy production. Fixed cells includes next type adipose cells, adipocytes, special cells of adipose tissue. In white adipose tissue, adipocytes has round shape. In central part present large fat droplet. Nucleus stay on peripheral part and has flat-like shape because large fat droplet will be removed nucleus to peripheral part. And in normal adipocytes form some groups in loose connective tissue. And in final, if we have a high amount of adipocytes, form and result white adipose tissue. Pigment cells, melanocytes, another name. Pigment cells has short processes, star-like shape. In central part of cytoplasm present melanin. It's a granules with pigment, and its cell produced in embryonic period from neural crest cells. Wandering cells include next subgroups. Mast cells, or another name, basophilic mast cells. Very large cell, 20-30 micrometer by size, derived from cells of stem cells of red bone marrow and characterized basophilic cytoplasmic granules. We will study in previous lecture and previous practical classes specific basophilic granulocytes, similar cell mast cells which derive in general precursor with basophilic granulocytes of blood, and in granules present histamine and heparin. Histamine and heparin very important in allergic reaction and in inflammation process. Macrophages, large cells, derived from monocytes and very active cell to migration to phagocytosis, and macrophages may be form some types of in different types of tissues. For example, in bone tissue represented by osteoclast. Osteoclast, it's a specific macrophage in bone. Or in other subtypes, you can see in your textbook and from previous lecture about subtypes of macrophages. On its slide, you can see mast cells after staining of hematoxylin and eosin, and in cytoplasm present more basophilic granules. In period of inflammation or allergic reaction, mast cells release granules and in result will be form an active process. Histamine provide forms of release plasma in tissue, and heparin protect from coagulation. On its picture photo from electron microscope, it's a macrophage. Macrophage has a large nucleus and more short processes which form process of phagocytosis. Macrophage, very active phagocytotic cell. Next type of wandering cells is plasma cells. Plasma cells form from B lymphocytes, and main function of plasma cell is production of antibodies, and plasma cell formed after differentiation, antigen dependent, and in its period, in its process, very active form T helper cell, and T helper cell helps to B lymphocytes, form specific B immunoblasts, and B immunoblasts, the next after will be differentiate and form active plasma cells. Plasma cells characterized by specific location of nucleus. Nucleus located more in peripheral part. In central part present light staining area, and inside present Golgi apparate. In plasma cell present in cytoplasm more rough endoplasmic reticulum which produce of antibodies, because antibodies is proteins. And in central part of a nucleus present specific picture which characterized of plasma cell. Picture form it's similar to screws or claw. On its slide present photo of plasma cells. You can see position of nucleus more peripheral part, and screws show light area in central part of cell, which locate of Golgi apparate. It's hem of plasma cell, nucleus specific picture present of heterochromatin, nucleolus in central part present, and slight area in central part of plasma cell include centrioles, you see, Golgi apparate. Rough endoplasmic reticulum present more in peripheral part of cytoplasm. Also present more mitochondria, because process formation of new proteins always energy dependent. Its slide represents to you functions of connective tissue propria, and all functions very important and include more special characteristics of combination of cells or location of cells. And for example, we have a mechanical support. Why? Because present more collagen fibers. Connective tissue propria provide exchange metabolites between blood and all tissues, because always loose connective tissue cover of blood vessels. We have rule of histology. When we have blood vessels, always surrounded by loose connective tissue in peripheral part. And appositive, if we have loose connective tissue in some part of organ, on some tissues include, always inside will be present blood vessel. Next, connective tissue propria participate or in homeostasis, instability of internal environment. Next, protection from infections, from different types of antigens. It's phagocytosis, production of antibodies in plasma cell. Next, repair after injury. Yes, of course, very important function, plastic function, because fibroblasts very active cells and produce more collagen fibers, and it's a fast case protection after injury. Next very important question, classification of connective tissue propria. Connective tissue propria is subparts of general connective tissues. I repeat to you from previous slides. Connective tissue propria include blood, connective tissue propria, cartilage, and bone. But connective tissue propria to divide on some parts, and in its case we divide on two main groups, fibrous connective tissue and specialized connective tissue. Fibrous connective tissue may be two subtypes. Loose connective tissue, which include not more fibers, more ground substance in extracellular substance. Dense connective tissue include more fibers and not more ground substance. And dense connective tissue to consent position of collagen fibers or elastic fibers to divide on two subgroups, dense irregular connective tissue and dense regular connective tissue. In dense irregular connective tissue, collagen fibers not form parallel layers, not form bundles. But in dense regular connective tissue, collagen fibers form bundles, lie parallel, and form very strong composition. Next, specialized connective tissues. Specialized connective tissues include specialized cells. More, for example, it will be adipose tissue, include more adipocytes. Reticular connective tissue include reticulocytes and reticular fibers. Mucoid connective tissue include more extracellular substance, ground substance, and not include fibers. And pigmented connective tissue include more pigment cells. Loose connective tissue includes network of different types of fibers, more collagen fibers, also elastic and some reticular fibers, include fixed and wandering cells, all subtypes, and very well vascularized. Also, I told you before, if we have loose connective tissue, inside will include blood vessels. It may be capilla, arterioles, venules, and if we have small vessels and large vessels, always its vessels will be surrounded by loose connective tissue. Next subtype, dense connective tissue. Dense connective tissue include more fibers. It may be collagen fibers. Collagen fibers form parallel layers in tendons, in specific junctions. And next, two types of dense connective tissue, regular and irregular, I told you. Irregular dense connective tissue present in dam of skin, regular in tendons, in some capsules which surround of some organs and provide protection function. Specialized connective tissue, it's a reticular connective tissue present in red bone marrow, in spleen, in lymph nodes, and include reticular fibers, another type sort of collagen, form delicate network and include reticular cells. Mucous connective tissue present in umbilical cord and include jelly-like composition of ground substance. And very interesting fact, in mucous connective tissue, not present fibers, just cells and amorphous substance. On its slide present schematical picture of composition reticular connective tissue. Reticular connective tissue present in organs of hematopoiesis, red bone marrow, spleen, lymph nodes, and reticular connective tissue include reticular fibers, black color on its picture, and reticular cells. Reticular cells form strong junction between one by one and always reticular fiber stabilized of its junction and form delicate network of organs of hematopoiesis. Adipose connective tissue is specialized connective tissue, and special characteristic of adipose tissue is a very high level and amount of adipocytes. Adipocytes, large cells, oval shape, and include specific droplets of fat which has more triglyceride. And adipose connective tissue surround of blood vessels too and always rich vascularized. Two basic types of adipose tissue in human organism. There are white adipose tissue and brown adipose tissue. White adipose tissue termed unilocular adipose tissue. Why? Because in adipocyte present large lipid droplet, and large lipid droplet will be form a position of nucleus in peripheral part of cell. Is large cell from 50 to 150 micrometer in diameter. It's photo from histological specimen after staining of hematoxylin and eosin, and you can see on slate light white areas, non-staining areas. It's an area of lipid droplet in adipocyte. It's fact formed after staining of hematoxylin and eosin because used in its process alcohol remove fat, and in result white areas or free areas in cell. Brown adipose tissue include more adipocytes, and adipocyte of brown adipose tissue has more lipid droplets, small lipid droplets, opposite of white adipocyte, and central location of nucleus in its cell, and uh, cell has a really brown color without staining because in cytoplasm present more mitochondria, in mitochondria present more cytochromes, and in result really brown adipose tissue has brown color. Main function of brown adipose tissue is thermoregulation, and present brown adipose tissue more in newborns, and until first year of life present more, and then after will be changed to white adipose tissue. On its scan you can see formation process of adipocytes, and precursor of lymphoblast and adipocyte is mesenchyme cell. Its scheme of brown adipose cell, you can see location of nucleus in central part of cell, more mitochondria present here, and you see white areas inside in cytoplasm. It's areas which include fats inside. And after staining of hematoxylin and eosin, visible white areas in cytoplasm, free, not without color. Its areas represent position of fat droplets. On its slides and next slides, you can see examples of test control exam, which include in test control exam you can use in your training, and on practical classes, on lecture, I will explain together with you correct answers to its test. Thank you for your attention, my dear students. I hope its lecture helped you to prepare to practical classes and to exam.